Welcome to Jinnious Lab. Today we will cover IB Biology B2.1.3-7 membrane transport.
So in B.2.1.3-7, we will be looking at membrane transport. So in summary, membrane transport can be subdivided into many classes depending on how they transport the molecule. The main types that we'll be looking at are simple diffusion, osmosis, facilitated diffusion, active transport, and this active transport can be further divided into primary and secondary active transport. You can always refer back to this table for a review. So we also have a diagram showing different types of transport mechanisms you can refer back to. So now let's actually move on and zoom in to each type of mechanism. So first of all, we'll be looking at simple diffusion of small and nonpolar molecules. So simple diffusion refers to this passive movement of molecules across the membrane bilayer along the concentration gradient. And this will happen until the equilibrium is reached. And because the molecules will be moving along the concentration gradient, we'll be calling it a passive. And by saying passive, it means that they do not require energy for the transport. And the meaning of moving along the concentration gradient means that the substances will move from the area of higher concentration of solutes to the area of lower concentrations of the solutes. And the simple diffusion will be the primary method used by the small and nonpolar molecules. And again, we know that cell membranes are semi-permeable, which means they will be allowing small and uncharged substances to diffuse across the membrane according to the concentration gradient. So small substances can actually utilize simple diffusion because they can pass through the lipid membrane directly due to their size. Also, nonpolar or uncharged substances can pass through the membrane directly because they are nonpolar, like the long fatty acid tails they need to go through. And examples of the small and nonpolar molecules would be oxygen and carbon dioxide. And then we'll be looking at the factors which may influence the diffusion rates. And first of all, the concentration gradient matters because if we have larger difference in solid concentration across the membrane, that means we'll be having a steeper concentration gradient and hence the faster movement molecules across the membrane. Second, particle size also matters because if we have smaller molecules, there will be less resistance experienced by the molecule, which makes it easier to pass through the lipid bilayer and hence leading to the faster diffusion rate. Then temperature also plays a role. If we have a higher temperature, we'll be having increased kinetic energy of the molecules and hence they will be moving faster. So their diffusion rate will be consequently be faster as well. And then we come up with polarity and charge of the molecule. And as we just discussed before, nonpolar and uncharged molecules are hydrophobic and hence they are lipophilic, meaning that it is easier for them to pass through the long fatty acid tails of the lipid bilayer. This leads to faster diffusion rate again. And then we can move on to the second type of the membrane transport, which is the osmosis of water molecules. So osmosis can be defined as the passive movement of water across the membrane along the concentration gradient of the solids. So again, it is a passive movement, meaning that they don't require energy or ATP to transport these solids, and they move along the concentration gradient. So, and also it is important to emphasize that this concentration gradient um, is referring to the solutes rather than the water molecules. Then let's move on to the mechanism of osmosis. So water molecules can randomly move across a semi-permeable membrane. And then there will be a situation where there is a concentration gradient of the solutes. In that case, water will move from the area of low solute concentration to high solute concentration until the solute concentrations on both sides of the membrane are equalized. So this essentially means the water molecules are moving from area with more free water molecules to less water molecules. And because of this, it is still a passive movement where the water moves along the solute concentration gradient. And then, even though water can actually directly diffuse across the cell membrane, we also have aquaporins involved in this process of transporting water. And aquaporins are the integral membrane proteins that serve as the channels for water to be transported across the hydrophobic lipid bilayer. And they allow the rapid and controlled flow of water in and out of the cell, ensuring that the cell can quickly adapt to osmotic stresses. So even though water can directly diffuse across the cell membrane, we also have aquaporins, which can facilitate water transport in a lot faster manner. So usually aquaporins are, like we mentioned, can adapt to osmotic stresses very well. And we'll be looking at cases that are related to this osmotic stress, where we'll be learning the concept of tonicity and effect of osmosis. And the effect of osmosis in plant and animal cells really depends on the tonicity of the environment. And tonicity really refers to the relative concentration of the solutes in the solution outside the cell compared to the inside of the cell. So we can largely divide the tonicity into three types: isotonic, hypotonic, and hypertonic. So in an isotonic solution, the extracellular solute concentration and intracellular solute concentration will be equal. So in this case, water will move equally in both directions. So it'll essentially that movement of water will be zero, which will allow the cell to be remaining in balance. And then we have hypotonic solution. And as the prefix hypo tells you, in this case, extracellular solute concentration will be smaller than the intracellular solute concentration. So water will enter through the cell, and this movement of water into the cell causes different happenings in the animal cell and in the plant cell. So in the animal cell, the cells will actually swell and burst. On the other hand, on the plant cell, we say that they become turgid. This difference largely comes from the fact that in plant cells, we have a cell wall, while in animal cells, we do not have the structure, only the membrane, which makes it more responsive to the movement of water. And then we have hypertonic solution. And again, as the prefix hyper tells you, in this case, we have higher concentration of solutes in the extracellular environment than in the intracellular environment. And here, to balance out the concentration difference, water will be leaving the cell primarily. And this causes the animal cells to shrivel, and this causes the plant cells to be going through plasmolysis. This refers to the process where the cytoplasm will be shrinking away from the cell wall, as shown in the diagram. Now we'll be looking at the third type of membrane transport, which will be facilitated diffusion. From now on, we'll be actually seeing how intermembrane proteins are involved in this process. So facilitated diffusion can be defined as the passive transport of molecules across the cell membrane facilitated by membrane proteins such as the channel and carrier proteins. And they allow the molecules that cannot directly diffuse across the hydrophobic core of the plasma membrane to move in or out of the cell. As we have discussed previously, we know the membranes are selective, so it is difficult for the polar or large molecules to go across the long fatty acid tails. And to help transport these molecules, we will be using channel and carrier proteins. So we'll be first looking at channel proteins, and they are integral membrane proteins that form hydrophilic channels across the cell membrane. So in this channel protein, we'll be seeing the arrangement of amino acids in a channel determined by their polarity. So we know that all proteins are composed of amino acids, and in the channel protein, polar amino acids will be positioned internally, oriented towards aqueous environments. And you can actually see this in the diagram on the left. And then we will be having nonpolar amino acids located externally in the channel protein, so they can directly associate with the lipid bilayer. So this structure essentially provides a pathway for polar and charged compounds to bypass the hydrophobic core formed by the long fatty acid tails of the plasma membrane. Then we can look at the carrier proteins, and they can be defined as integral membrane proteins that change conformation to transport the substrates. So unlike the channel proteins, which looked like a tunnel and they did not change conformation, in carrier proteins, we'll be seeing the conformational changes. And they will be remaining open at one side of the membrane at any instance. And then when the ions fit into their holes, they can carry ions or molecules across the membrane via conformation change, like shown in the diagram. Now we can move on to the active transport. So in facilitated transport, we mentioned that it was a passive transport, meaning that it did not require use of energy in the form of ATP. But now, when we talk about active transport, we will be needing the energy ATP. So active transport can be defined as the process where cells use energy, typically in the form of ATP, to move molecules across the cell membrane against their concentration gradient. So the fact that they use ATP allows them to move against their concentration gradient, which are not favorable. So active transport can be further divided into two classes, which are the primary and secondary active transport. So primary active transport uses energy directly derived from ATP hydrolysis, and secondary active transport will be using energy indirectly derived from the concentration gradient of the other solute. So let's look at the primary active transport first. So here, what happens is that ATP will bind to the protein pump to activate it, and then ATP will be going through hydrolysis, and in this process, the protein pump can gain energy to transport the solids against their concentration gradient. And then in primary active transport, we can see that this protein pump plays a big role. And protein, when we utilize protein pumps, we can see a specific solute binding to the protein pump on the one side of the membrane, and the conversion of ATP to ADP and the phosphate group, which we call ATP hydrolysis. And this process will actually cause a conformational change in the protein pump. And this change will move the solids across the membrane against this concentration gradient, and then release the solid on the other side. And then we can move on to the secondary active transport. So in this case, we won't be using ATP, but we'll be indirectly using the energy from other molecules, which will be moving down its concentration gradient. So it is best to talk about secondary active transport with an example. And an example would be sodium glucose co-transporter. So here we have sodium, and sodium have higher concentration outside of the membrane than inside of the membrane. And hence, sodium ions can move down the concentration gradient from the extracellular side to the intracellular side. And this movement of sodium ions can actually lead to the movement of glucose against this concentration gradient as well, because the sodium ions, when they move along the concentration gradient, they will be simultaneously bringing the glucose molecules against its concentration gradient, like shown in the figure 8. And the secondary active transport can also be further subdivided into symport and antiport. And in symport, we'll be seeing the transport of two substances in the same direction across the membrane, like how we just saw the example of sodium and glucose co-transporter. And in the case of antiport, we'll be seeing the transport of two substances in opposite directions across the membrane. So it is important to remind you that both in symport and antiport, one of the substrates move down the concentration gradient, which will be providing the energy to transport the other substrate against its concentration gradient.
For IB45 Genius, Jinnious Lab, don't forget to subscribe and like.